will see on that study guide, typically I put all the diagrams up for you to print off, but I don't have them. So the diagrams are listed. They are diagrams from your lab book, okay? But I don't have copies of unlabeled diagrams. Luckily, because we use a lab book that accompanies our textbook, I'm able to have those diagrams from the files I get that I use in lecture. And there's an option to have them unlabeled, so I put them up there. But I don't have images from the lab book, okay? And basically they're muscles. The muscles are a pain, but there's a list there, yes? Yeah, so there you go. You're not getting a word bank, but there's a list, and it's short. So no need to panic. No panicking. You just don't know which diagram you're going to get to label that muscle. So you should be able to find those muscles on the diagrams if they're present. Okay, and then mainly, most of the, as far as fetal pig goes, the diagrams that I put up there, the color ones that I reproduced from the labs, the ones that hopefully you use when you do your labs, any of those organs. Anything that's visual, I can label with a pen is fair game. Okay, you're going to have two pigs. One's going to be what? One's going to be a girl and one's going to be a boy. So you should be able to label both parts in males and females as far as reproductive system goes. And you should be able to label all those parts. So take time in lab to know that before you leave. Because these labs can go pretty quick. And I do. I have some students that come in there and say, oh, yeah, I'm out of here. I'm going to go enjoy the sun. But when it's time to take your test, you might not be happy that you made that choice. So take the time to be able to identify those parts. Take the time in lab. Okay? All right. So let's hit it. Where are we going? So we left off chapter 26. A lot of this information is repeat information. Have you figured that out already? So I'm not going to take tons of time in this chapter to explain the concepts again. You should know the concepts by now. Yes? We talked about a lot of this stuff when we talked about hormones. We talked about a lot of this stuff when we just finished the renal system. So I'm not going to take tons and tons of time on this chapter to retell you the same information. You with me? But I am going to tell you what's important. So this diagram here talks about, and we can see this guy on page 999. This diagram, it's a girl. On page 999, figure 26.5. And this talks about regulation. One of the things I want you to take advantage of in this chapter is the diagrams. Because if you can follow along with the thought process in the diagrams, then you've got it. Yes? And basically what this chapter talks about is fluid regulation in fluid compartments. So when I talk about plasma osmolality, what is that referring to? So concentration of dissolved substances in our liquids. And they create what we call osmolality. When osmolality increases, what's going on with those fluids? They're starting to become more concentrated. There's more dissolved substances in them than the water balance there should be. So in order to compensate, fix that, what do I have to do? Got to get more fluid into your body. So we have mechanisms, receptors that are going to monitor blood osmolality, and guess where those Osmo receptors are located? Hypothalamus. Everybody understand what an Osmo receptor is? What is it? Very good. It's a receptor. What's it recepting? It's measuring the concentration of dissolved substances in fluid, osmolality. And they're part of the central nervous system. Remember, we have tons of receptors in the central nervous system constantly monitoring levels of a whole bunch of different things. Blood glucose levels, blood sodium levels, water levels here, and osmolality, an indirect measure of water levels. So receptors are firing off. They're telling you, you need to get more water into your fluid compartments. And what are you going to start to do?
Thirsty.
What's thirsty feel like? Your mouth starts to get very dry. So you're going to decrease production of saliva. And what do you want to do when your mouth starts to get very dry? Go get a drink. So hypothalamic thirst centers are going to then fire off, and this series of changes are going to happen in your body because you need to get more water in. So water's going to come in. Hopefully you'll go get a drink somewhere. You'll moisten the mouth, the throat, stretch the stomach, stretch the intestine. Water gets absorbed fairly quickly into the system. Okay, and you actually start eliminating, say I go and have a big, huge drink of water. When do you start eliminating that water that you had a big, huge drink of? Pretty quickly after you drank it, if it's too much. So about even up to an hour after you have a big drink, you're going to start eliminating excess water. So that goes into your circulatory system very quickly, unlike some of the other things that are absorbed through the gastrointestinal tract. The other thing that happens when plasma osmolality goes up is plasma volume tends to go down. And we know that that's a problem as well, right? What are the kidneys' fluid balance mechanisms going to do to try and increase plasma volume? Yeah, we're gonna get some help, direct help, from those cells in the nephrons that are doing the absorption and secretion from the renin-angiotensin mechanism. Yes? And they are going to cause what to happen? If I'm thirsty, my osmolality is high, my water levels are low, what is the kidney's role going to be in this? Reabsorb more water back into the circulatory system. And that's going to help regulate the pressure imbalance from an increased plasma osmolality. See, you know all this stuff already. Yes? Say yes. Say yes, I know this. I'm not getting a comfortable feeling about yes, you know this. So again, use your diagrams in this chapter to your advantage. So regulation of water intake then is very important, and different hormones like renin and angiotensin have an effect on the renal system to help balance water in the body. But other hormones are also going to have an effect on the renal system to help with water balance as well. Again, increased osmolality means increased concentration of dissolved substances, means increased concentration of sodium, for example, in plasma. So we also have hormones that are going to help affect tubular reabsorption and secretion, and that whole kidney mechanism we discussed in the previous chapter, like antidiuretic hormone. What's antidiuretic hormone doing for me? Hold on to water. How? It's going to help me hold on to water, but who's actually holding on? Yeah. When we talk about reabsorption, remember where we had the whole process of absorption, secretion, kidney, nephron, those three different things that the nephron does for a living. When I talk about reabsorption of water, what actually am I reabsorbing? Sodium and water follows it, exactly. So antidiuretic hormone is going to have a direct effect on the collecting ducts of the kidneys. And we are then going to have an increased water reabsorption and decrease osmolality, again, increasing plasma volume. What's going to happen to the amount of urine you make when you are in water conservation mode? It's actually going to become more concentrated. If I'm trying to conserve water, I'm going to reabsorb more water back into the circulatory system. The waste still has to move through, and that's why they talk a little bit about regulation of water output and how we can't go very long without water. Can we? We can go a long time without food, but we can't go as long by any means without water, because we're constantly filtering our blood supply no matter what. Urine will become much more concentrated and much less in volume when I'm in water conservation mode, but you're still going to lose it. Okay? And you can't go as long without water as you can without food. Disorders of water balance, dehydration, hypotonic hydration, edema, all talk about mechanisms that you kind of already know about. Remember when we had our discussion way back when, and I can't remember what chapter it was, probably 3? You remember chapter 3, okay? When we talked about tonicity, and we talked about dissolved substances in our fluids. Well, when we talk about ECF, what's ECF? Extracellular fluid, there creates a pressure gradient due to the amount of dissolved substances in those fluid compartments. And when you become dehydrated, what happens to that osmotic pressure in extracellular fluid? It's going to start to rise. And what's going to happen to our cells? They start to shrink. How come? Yeah. And if you want to kind of think of it tonicity words, the amount of dissolved substances in that fluid on the outside of the cell is higher or lower than the fluid on the inside of the cell. Or it's going to be higher. So water is going to do what to try and compensate? It's going to leave the cell, and that's why the cell shrinks. How about in hypotonic hydration? Can you kill yourself by drinking too much water? Yes, you can. It's hard to do because a lot of our central nervous system mechanisms will stop us from doing that, especially the stretch reflexes in the stomach. But can I do it? Yes, I can. And what happens? Yeah, and your cells will do what? Swell to the bursting point. So that extracellular osmotic pressure is going to fall. It's kind of the opposite of what we keep talking about when we talk about pressure changes. But think of it in terms of tonicity. What happens to the concentration of dissolved substances outside the cell? It becomes less than inside the cell. Where is the water going to go to even out? Inside the cell. Yes? Okay, so dehydration versus hypotonic hydration. What's edema? And what's that caused from? Yeah, when we start to have ion and buffer imbalances and fluid concentration imbalances, we start to have buildup of fluid in extracellular compartments, interstitial fluid. And there becomes a situation where it's hard to compensate for that extra fluid that's outside the circulatory system, for example. Who helps me reabsorb fluids back into the circulatory system? Besides the circulatory system itself, the lymphatic system. But remember, things move with a pressure gradient, right? So if I disrupt the pressure gradient or I disrupt the ion balance, and that's another reason why water might move, right? To follow ions, that obligatory water reabsorption, I might have problems with buildup of fluids in interstitial spaces. One of the things we're going to talk about in this chapter is the role of proteins in keeping fluids in their compartments properly. And when I start to decrease protein intake drastically, starvation, for example, and you've probably all seen these terrible images of these poor children in third world countries who are starving but have a very large abdominal pelvic cavity. They have huge bellies. Why? Yeah, because they're so protein deficient, their fluid compartments start to fill with fluid. So that's not their stomach full of food, that's their abdominal pelvic cavity full of way too much abdominal pelvic fluid because of protein imbalances. So the balance of ions in our fluid compartments are going to play a big role in keeping the proper amount of fluid in those fluid compartments. Yeah, once you start closing down systems to compensate for those fluid imbalances and they start to become damaged, there's sort of a point of no return. But a lot of those children, that's a reversible thing. It takes a long time. But good nutrition is important, right? Nutrition class, spring, next month. So, the next thing the book goes on to talk about is the role of those electrolytes and the role that they play in helping to keep those fluid compartments balanced. Causes and consequences of electrolyte imbalances. Excuse me, table 26.1 on page 1003. Look at all the information you have in that brain. It's full. It stops at 1,000. Talks about some of the different causes from decreases or increases of some of these electrolytes or charged particles in our fluid compartments. So I want you to just basically have a general definition and understanding of that table. So when I say hypernatremia, you're going to know that it's what? Too much what? Why am I calling it natremia? Oh, chemistry people in the classroom. What is the atomic symbol for sodium? Na. Why is it Na? Yeah, some Latin or Greek or one of those things. Natrium is sodium. Okay? So, hyper or hyponatremia, hyper or hypokalemia, which is potassium, hyper or hypophosphatemia, and phosphate is also a very important electrolyte that helps keep fluid compartment balances. Chlorine, calcium, magnesium, also very important ions that not only play roles in very important reactions of metabolism, but they also play very important roles in helping to balance fluid compartments in the body. So everybody good with that? General definition. Regulating sodium imbalances and some of the mechanisms of and consequences of some of the different hormones that are going to help with that. Aldosterone, for example, is going to help sodium, potassium, ion balance. And again, when I balance my ions, what else am I going to balance? Well, pH is fluids. So what kind of, the chapter's starting to repeat itself. You notice this? Just saying the same thing, a whole more chapters, a different way. Somebody, the fluid and electrolyte guy has to feel important. So ANP is another one, and of course we know renal cortex, aldosterone, It's going to have an effect on the kidney tubules. It's going to cause increased sodium what? Reabsorption, which is what? Going to lead to what? Increased water reabsorption as well. Don't forget our friends renin and angiotensin. And then ANP. What's ANP? Atrial something peptide. Atrial natriuretic peptide. And this is going to be released by cells up and around that vena cava region when blood pressure starts to, well, actually aortic region, I'm sorry. When blood pressure does what? Look at the decreased stretch of the atria is going to cause the release of ANP. And what are the effects of ANP? What do I have to do to decrease pressure in the system? We've been talking about increasing it forever. Now I got to get rid of more water so that I can decrease pressure in the system. So I'm going to decrease the release of some of these guys like ADH because I don't want to hold on to more water. I want to do what? Get rid of more water. So I'm going to decrease the release of things like aldosterone. And then what's going to happen in those nephrons? Look under the kidney where it says effects. That's right. I'm going to decrease the reabsorption of water and therefore decrease the reabsorption of, decrease the reabsorption of sodium, therefore decrease the reabsorption of water, therefore, there you go. Decrease blood pressure. Yes? Decrease blood volume. I want to say it out loud so when you go back and listen, you'll say, what the heck did you? What's she talking about? You get it? You understand? Yes. Get it? Got it? Good? Influence of other hormones. Oh, here we go with more diagrams. Female sex hormones, estrogen, progesterones, are going to have an effect on reabsorption of sodium as well. If I decrease sodium reabsorption, what's that mean? If I decrease sodium reabsorption, I decrease water reabsorption. I'm going to pass more water where? Out as urine. Yes? Glucocorticoids. Going to help me do what? Tubular what? Tubular reabsorption. Enhance tubular reabsorption. Everybody understand what that means? Regulation of potassium, cardiovascular baroreceptors. Before we get too far past that, again, if I see that the pressure in the system, these baroreceptors are monitoring pressure in the system. If pressure in the system goes too high, what am I going to do? Try to lower it. What do I have to do and what can I do fast? Because we're talking about fluid compartments, right? Pressure in the system goes up. I want to bring it down. What do I have to do? Get rid of more water. To get rid of more water, I need to get rid of more sodium. Yes? Regulation of potassium balance, cortical collecting ducts, influence of plasma potassium, influence of aldosterone, repeating yourself again, textbook. We understand what they all do, right? Hopefully. Mechanisms regulating sodium and water balance help maintain blood pressure homeostasis. And this is kind of the mother of all the diagrams in this chapter. Kind of ties everybody together. Tells us basically we're affecting kidney function when we try to regulate water compartments. Again, I've said this way too many times, right? So if I have a decrease in systemic blood pressure, that means I have a decrease in blood volume. What do I have to do? Increase blood volume. This is, again, tying all the stuff we just talked about all together in this one diagram. Repeating myself. All right, so regulation of calcium and phosphate balance is also important, not only for regulation of fluid compartments, but they also give us a little blurb about what other systems rely on these ions and ion balance for good health. So things like bones, for example. We know what parathyroid hormone does for us, right? What does it do? It does lots of things. Give me an example of all the lots of things parathyroid hormone does. Activates bone digesting osteoclasts. What's that doing for me? Say who? Oh, sure. Getting calcium where? From the bones into the circulatory system for transport. So when calcium levels go down, I'm going to start wearing away my bone to bring calcium levels back up. Give me some of the important things calcium do for you in maintaining homeostasis. Who? Blood clotting. Muscular. Somebody said neuromuscular. Nerve impulses and muscle contraction, to name just a few. Yes? Excuse me. Small intestines. PTH is also going to enhance intestinal absorption of these substances as well. So remember, sometimes hormones play dual duty as far as activating cells and what they do for a living. So if I am secreting a substance that's going to enhance my absorption or reabsorption of a substance like calcium, it also may affect other organ systems that are responsible for absorbing calcium as well, including the small intestine. And then, of course, the kidneys. Parathyroid hormone increases calcium reabsorption so that I don't lose calcium where? In my urine. Yes? Okay. Anions and cations. What the heck are those? Negatively charged ions, what do we call them? Anions. Positively charged ions, cations. They talk about regulation of anions and why they're so important to maintain fluid compartments. Again, think about water. It's a polar molecule, and it's going to follow these charged particles. So their concentrations are very important. Big anions like proteins, we just mentioned a moment ago, play a big role in helping to keep those fluid compartments balanced. So when I start to decrease the amount of anions, including those big negatively charged proteins, I'm going to start messing with the fluid compartment balance. This is a beaker with some important stuff that's also going to be a major player in reactions of metabolism throughout the body. And we're revisiting our information from way back when in Chapter 2. Acids and what? Bases. What's the definition of an acid? Yeah, contributor of hydrogen ions in solution. The more hydrogen ions in solution, the more acidic the solution. What's the pH do? Remember that? Confusion? So a pH below 7, which is neutral, and this is the place where we have what? There's two ions that are going to influence pH. The ion pH definition is based on hydrogen ions, but there's another guy that also affects pH. Who is that? Hydroxide ions. Ringing a bell? OH or hydroxyl ions? So in our bodies, we're constantly trying to maintain the proper pH. What's proper body pH? You're dead. 7.35 to 7.45. So we have something called buffer systems that are constantly either contributing or taking up excess ions to help maintain the proper pH in our body fluid compartments. So acid-base balance is extremely important for things like protein integrity. If I mess up the pH in my body, what am I going to mess up with respect to proteins? You know, proteins are made of amino acids. I'll give you a hint. Think about your experiment on the digestive system in lab. Now you did this with temperature, but you can also do it with pH. You took some enzymes, which are proteins, that you were going to use for digestion. And you took them and you put them in a tube, and then you took the tube and you stuck it in boiling water. How'd that enzyme work? Did it work? No. It did what? I heard it. It denatured the enzyme. It broke apart the protein. pH can have the same effect. So buffer systems, and they are highly dependent on ion concentrations in fluid compartments throughout our body, are constantly going to make sure those ion concentrations are balanced. So they talk about chemical buffer systems, bicarbonate buffer systems, which is just one of the chemicals constantly trying to maintain pH through the system. Am I going to make you memorize these chemical things? No, but you should understand that bicarbonate buffer systems, and what bicarbonate is, it's H2CO3, are going to help to balance pH in the system. They also talk about phosphate buffer systems and protein buffer systems. Again, all of these different buffer systems, and believe me, these are just the main categories. There's tons and tons of different ones. But the gist of the information I want you to understand is these buffer systems are constantly either taking up excess hydrogen ions or contributing hydrogen ions to our fluid compartments to make sure pH balance is maintained. pH balance is important for reactions of metabolism. If I mess up pH in one direction or another, I'm going to mess up chemistry big time. And we talked a little bit about this when we talked about the respiratory system. If I have too much carbon dioxide, I'm going to make too much what? Carbonic acid. Is that ringing a bell? If I make too much carbonic acid, what's going to happen to my pH? It's going to go down. It's going to become more acidic. So acidosis. And we can have acidosis or alkalosis, the opposite, not enough carbon dioxide, because of respiratory issues, but we can also have imbalance in pH because of metabolism. So they talk about renal mechanisms for acid-base balance, and we talked a little bit about this when we talked about tubular secretion. Near the end of the nephron, we're going to kind of fine-tune the mix before we throw out all that urine. And when I say fine-tune the mix, I'm referring to either absorbing more or secreting more hydrogen ions. And what's that going to help me do? Excuse me. What's that going to help me do? If I have too many hydrogen ions in solution, I have to do what? Get rid of them somehow, right? Either bind them up with a buffer, neutralize them, or get rid of them. And that's what these, way too complex diagrams are basically showing us, is how we are going to, this one says reabsorption of filtered HCO3 is coupled to what? Hydrogen secretion. So this is our chemical way of getting rid of some excess hydrogen ions. And where do you think this is? You have the yellow stuff over there on that side. Yes, that's urine. So what do you, who forms urine? The kidneys. What part of the kidneys? Well, that's where it collects. But who does the work? The nephrons. So these are actually looking at cells in the nephrons. And what are those blood vessels called? Peritubular capillaries. Okay? You with me? So these mechanisms, and I'm not going to make you memorize them, but they're explained in the book, are different ways of balancing hydrogen ion concentration, therefore balancing pH in the body. So these are the mechanism of action of some of the buffer systems and the kidney's role in helping to balance pH. Abnormalities of acid-base balance. When pH starts to get messed up, too many hydrogen ions, not enough hydrogen ions, both of those things are a problem, can be due to the respiratory system. Problems with getting enough oxygen in and getting rid of enough carbon dioxide. You know this already, right? Respiratory acidosis, respiratory alkalosis. The pH can also mess with the receptors and mess with breathing rate and heart rate, right? Because that's how the receptors know that your pH is starting to be off. So hypoventilation, hyperventilation, those are terms you know, right? When the pH goes too much in either direction, either respiratory or metabolic acidosis or alkalosis, systems are going to start to shut down. And they are trying to do what when they shut down? Well, they protect, it's a protection mechanism. If they work too much, they're going to make too much waste. They're going to contribute to the problem. They shut down for a little while. Maybe things will get better. Unfortunately, some of those systems are very important to keep you going. So respiratory system, we know its role in helping to maintain pH, but the renal system is also going to have a role in helping to maintain pH as well. Kidneys, ureters, bladder, urethra, all of those wonderful little electrolytes, and some of the consequences of acid-base imbalances. Just a general knowledge. Yes? We're done with chapter 26. Chapter 27. Now, chapter 27 is going to talk to us about the reproductive system in both males and females. And what is the goal of the reproductive system in humans, whether it be male or female? To reproduce. Or to make what? In order to reproduce, what do I have to be able to make? And what do we call sperm and egg, or its proper name is oocyte? They're different cells than the rest of our body. They're called reproductive cells, and they're a little bit different. Now we have to go way, way back and visit, I think it's chapter 3, when we talked about a process of cell division. Do you remember way, way back when we talked about mitosis? And what was the purpose of mitosis? To create identical cells from a parent cell. And mitosis is going on all the time in our body, right? Give me a place right now, a system that's creating new cells. Skin, epithelial cells, everywhere. They're reproducing like rabbits. Yes? And we're making copies of those cells. When we talk about the reproductive organs in mammals, or sexually reproducing organisms, the cells that are made to make a new organism are a little bit different. They're not exact copies of your cells. And when I talk about copies, think about the map that makes you who you are. DNA. How many or how much map do you have? When I talk about your genetic material, I'm talking about your DNA. That's your map. That's where I get the information to do what?
Reproduce yourself.
Make you. What are you made of? Huh? Cells and, yeah, and what else?
Organ systems, water,
And your DNA, your map, what's its job? Making proteins. Proteins that make you who you are. You, your skin color. You, your eye color. You, your hair color. Those are all protein in nature. And where do I get that information from? The map. You have 46 chromosomes worth of information. You actually have 2 bits of information for every protein that you can possibly make. You have 23 pairs of chromosomes. Yes? And where did you get your bits of information from to make you who you are today? You got those bits of information from your parents. But you only get half from one parent and half from another parent. You see where I'm going with this conversation? So I have to be able to, in my reproductive organs as a human, make cells with what? Half the information as all the other cells in my body. When we talk about heredity, we're going to talk about how this is done. But what the reproductive system's goal is to do is to, A, make those reproductive cells. Create cells with half the genetic information in them, and then deliver that information so that those cells can eventually come together and do what? Make a whole brand new organism. Now in the males, there are special portions of the male's reproductive system responsible for maturing those cells, and then there are portions of the male's reproductive system that are responsible for delivery of those cells. Yes? So, when we talk about the anatomy of the male's reproductive system, it's definitely going to be a little bit different from the anatomy of the female's reproductive system. What's the difference between the male and the female in human? Male's delivery system, females an incubator. So she also had, no, we don't sit on them. They actually develop on the inside. 40 long weeks it takes us humans to make a brand new one. But the two cells, those reproductive cells, one created in the male and one created in the female, will come together in the female's reproductive system and will incubate or grow from a ball of cells. And how many cells do you come from? You come from how many cells? One created by two what? Two half cells, kind of, to create one brand new cell. Well, that will then reproduce itself a bazillion times to create you as an organism. So the female's reproductive system not only has parts in it to create those cells, but also has a place for the new organism to develop and mature. So that's where we see the differences in the male and female reproductive system. So where in the male's reproductive system are those cells matured? Well, they are matured in an area called, or they're created in an area called the testes, and they are matured in an area called the epididymis. And where are these cells located in the male's body? They're on the outside, believe it or not, in the scrotal sac. So these cells, the beginnings of these cells, are created in the testes. They are then delivered out of the male's body, and they have to kind of make their way back into the male's body and then back out again through several other different parts of the male's reproductive system. So the book starts to talk then about the testes. And the testes, again, are the portion of the male's reproductive system where these cells are born. They come from immature cells or stem cells located in the testes. And they are going to have quite the travel before they actually make it out of the male's body. This is going to allow them to mature and get up enough energy for their long trip to come. Because once they're delivered into the female's reproductive system, they have to then travel up the female's reproductive system to find the female's reproductive cell in order for fertilization to take place. So all of these structures in the male are designed to mature and strengthen those cells for delivery into the female's reproductive system. So in the testes, we're going to find a series of tubes called seminiferous tubules where these cells are born. In the male's reproductive system, the most ideal temperature for the birth and maturation of these cells is actually lower than body temperature. And that's why the scrotal sac that contains these structures is outside the male's body cavity. If I take a look at the inside of these tubes, I'm going to see the cells. Oh, crack, crack. Did you hear that? Jeez, I'm getting old. Spermatogenic cells in tubule of epithelium. These are the immature cells that line the tubes, seminiferous tubules, in the testes. And these are the cells that are going to mature to eventually become the reproductive cells that will then take their travels through these tubes, through the lumen of these tubes, and eventually out of the male's body. You can kind of see, because you guys are all amazing histologists at this point, second semester anatomy and physiology, right? What's that cell doing? Yeah, what phase is it in? Phase 2. It's reproducing itself. It's condensing its genetic material. It might possibly be in prophase. Right. Very good. Prophase. Yes? Yeah, it was kind of hard to see that. I'll help you. But those cells are starting to reproduce themselves. When we talk about the process of making reproductive cells, it's very similar to what we've already studied when we talked about mitosis. Remember when we talked about mitosis, in order to make a brand new cell, and the cell kind of hanging out doing its job for a living is in something called interphase. Does that ring a bell? Okay. When it gets ready to start the split and make a brand new 2 cells, daughter cells, what does it have to do? It's got to first of all make a copy of the map. And that's going to happen during S phase of interphase. Is this ringing a bell? So you have to remember this crap. During S phase of interphase, I'm going to make a copy of the map, and then I'm going to go through the mitotic stages of my life. Anybody? Prophase, metaphase, anaphase, telophase. And I'm going to make 2 new daughter cells. When we talk about making reproductive cells, we're going to do that. Those immature cells, the spermatogenic cells, are going to start to do that. But what happens next to the 2 new daughter cells is we don't give them a chance to make a copy of the map again. We're going to send them into the mitotic stages fairly quickly after they're born. If I don't get to make a copy of the map, the 2 cells that come from my split, and this process is called meiosis, are not going to be exactly the same as the parent cells this time. They're only going to get what? Half the information. So when we talk about human chromosomes, that first split of mitosis, I'm going to go from 46 chromosomes to 92. I'm going to split. Then those 2 new guys are going to go right into the mitotic stage again. Meiosis. I'm going to split. Now I go from 46 chromosomes to 23. So the result, after 2 stages of splitting in meiosis, and in meiosis we call it meiosis 1, which is pretty much the same thing as mitosis, and meiosis 2, which is the same thing without making a copy of the map, we're going to end up with, from one of these immature cells in males, we're going to end up with what? 4 reproductive cells. What's different about them? Do they have 46 chromosomes? How many do they have? 23. There's my half cell. There's the male's contribution. You with me? Again, we'll talk about meiosis again. That's kind of near the end of the chapter. But that's kind of the gist of it. It's the same as mitosis, except it happens twice. And the second split, I'm not going to give you enough time to make a copy of the map. So that the end result is 2 half cells, or 4 half cells in this case, from the first immature cell. So they're going to give rise, or keep turning, down here in the linings of the seminiferous tubules that I find in the testes. So this is where the sperm are born, and that's what we call the male's reproductive cells. In order to then leave the male's body for delivery into the female's body, the sperm that were born down in the testes have to mature. And what maturation for these cells means is I have to surround that genetic material that I'm going to deliver into the female's body with some protection. And I also have to provide that cell with some way to be motile. Now, most of the cells in the human body are not motile. You know what I mean by motile? I can move from what? One place to another. Except for these guys. So what do I have to give these guys in order for them to be motile? A tail. let's get scientific now. It's not really a tail. It's called flagella. And flagella is going to allow these cells to move through the female's reproductive system after they've been deposited to find that oocyte or egg for the fertilization process. Millions of these wonderful little cells are produced for delivery in males at any given time. Why millions? Millions, like 2 million. What are the chances of making it to their goal? Slim, because the female's reproductive system sees them as foreign. So, if I make millions of them, the chances of them making their goal are better. So when we talk about maturation or spermatogenesis, it again is going to begin in the testes and then travel through many different accessory organs in the male's reproductive system to make it to the female's reproductive system. So the book talks about the testes and the seminiferous tubules and the penis, which is the delivery system into the female's reproductive system. Again, many of males' not only development areas but delivery systems are found outside the male's body cavity. But when we talk about the female, it's got to be in more of a protected environment because they are also what? Yeah, they're also the incubators as well. So we're going to find a lot of these female reproductive organs internal. But delivery system for the male is the penis. And the urethra in males is shared not only with the urinary system, but with the reproductive system as well. All right, are you ready to blow up and explode? All right, we'll pick up with the male's duct system on Thursday. And I hope to finish this chapter on Thursday and possibly even start the next chapter. Yes?